Thank you everyone for joining us today for our second Lobular event and I guess our first official Front Range ILC event. My name is Matt Sikora. I'm assistant professor in pathology and scientist studying lobular breast cancer. I'm really happy to be joined by my lab and by our great advocate team that's kind of forming the genesis of Front Range ILC. Before I get started, huge thanks to the Breast Cancer Care and Research Fund for supporting our event, making both the past one and this one possible. And huge thanks to the University of Colorado Comprehensive Cancer Center for hosting and providing logistic support for setting this up. So as I mentioned, we had our first event back in June 2022. We did over at the Night Horse building on campus, and it was presentations by Tracy Cushing from the patient experience, Dr. Elias from the clinical experience, and I talked a little bit about where the lab was going. This is on YouTube, and I think we have 400 some views, which is not a lot, but pretty exciting for us. But it's been a busy 10 months since. Huge thanks to Lori, Tracy, Donna, and Sue for helping move this group forward, both the heavy lift of kind of coalescing the group that came to that first June meeting into what we're now calling the Front Range ILC group. Lori, Tracy, and Donna were huge helps getting a new lab and Front Range ILC newsletter off the ground, getting articles written, getting the posting set up, and Donna and Sue are now my new MetaViver grant team for my first application to a metastatic foundation. So that's really exciting. So I think it's been a pretty busy 10 months, and here we are. What I hope is the first steps of this group coming together and really pushing lobular advocacy in the region forward. Before we get into it, though, I do want to bring up, and this is in memory of Lee Pate, and really this all started because of her. I was picking through emails and found this one back from January last year, where Lee emailed Tracy and I about the microgrant program at BCCRF, suggesting that maybe we would want to do a community outreach event. We had that first event June 1st, and Lee passed later that month. So I'm really gratified that I was able to share with her how successful that first event was. But really, this is all because of her. But even after she left, Lee has been a huge benefit to the lab. She and Lori initiated the Lobular Breast Cancer Research Fund, and Lee left a very generous gift in her estate. And so I'm really pleased that her name's going to be on our benefactor wall soon. And it really means a lot, I think, to all of us that her name's going to be attached to this program moving forward. So thanks, Lee. All right, the program. So we're going to talk while everyone's eating lunch a little bit about what research is going on, give you kind of a high-level view of a couple of the specific projects on lobular cancer biology in the lab, really background noise while everybody's eating lunch. Also that's going to be by myself and Dr. Maggie Music, a postdoc fellow in the lab. I'm going to hand off then to Tracy Cushing and Donna Charlevoix, who, along with Ann Bell, who couldn't join us today in person, are going to, I think, take the lead with really pushing this group forward, and they're going to talk about some of the ideas they have for how this group can advance advocacy in the region. That'll kind of be an introduction to the next few conversations. Lori Petiti is here from California, the president of the Breast Cancer Care and Research Fund, and my longtime advocate partner on at least 3 different grant proposals that are funded. And Lori's going to talk about roles that patient advocates have in the research and development process. Then we're going to hear Jen Lowry from the University of Colorado Cancer Center is going to join us online. She's from the Office of Community Outreach and Engagement at the Cancer Center, and is going to talk about how the Cancer Center is trying to put together a patient advisory council, and I think how this group can play a part in the development of that group and how they move forward. I saw Sienna Kostner is online from the American Cancer Society, and she's going to talk about how we can work with the American Cancer Society North Region. They've been huge supporters of my research, and we're really eager to hear how they can help support our advocacy efforts as well. And then we're going to hand back off to Tracy and Donna and open up to all of our partners in the room and online to talk about how this group wants to move forward. Really, my goal is to talk about research and then get out of your way, because the rest of that is we're just the lab rats. We're just doing the wet work, and the rest of it, we're eager to hear how we can support you. So with that, I'll just launch right into the research. I'm going to talk about one of our particular projects. You'll learn more about this in the next Sikora Lab newsletter. Donna did a great write-up of our R01 project that just got funded, that you're going to hear more about. But I'm going to talk about how some new funding in the lab is helping us understand how molecular partners of the estrogen receptor change its function, how we're using that information for developing new treatments for lobular breast cancer. All right, so before I launch into the science, I make it clear that I don't actually do anything in the lab anymore. I just write the grants and talk to you all about the science. My lab is here today. So background on all of us. So I'm from Michigan. I got my bachelor's and doctorate from the University of Michigan. Here is Dr. Maggie Music, got her undergrad in Tennessee and then did her PhD at Clemson. She's going to talk to you about her work on lobular cancer tumorigenesis, or understanding the earliest stages of breast cancer formation in lobular. I'm going to talk myself about Joe Sotnick's work. Dr. Sotnick is local, he's from Colorado, did his training at Colorado State. He does everything related to estrogen receptor genomics, proteomics, and really everything else in the lab. And then Maddie Shackelford is from our lab technician. She does pretty much literally every project in the lab. She's from Washington, did her undergrad training at University of Hawaii Hilo, and studies cell biology and lobular cancer and actually how it interfaces with similar biology in ovarian cancer. And we are nothing without our funding. And so huge thanks to the NCI, the Department of Defense, Dynami Foundation out in Michigan, Cancer League, American Cancer Society, and a lot of their breast cancer research fund. So folks online too, please feel free to chime in with chat or just unmute and ask questions. We'll have some time for Q&A too, but please ask questions along the way. So my lab is pretty much everything we think about circles around the estrogen receptor. And I'm sure all of you patients know your estrogen receptor status, but what is this estrogen receptor thing that you hear so much about, whether it's positive or negative in your tumor? So this is a sample immunohistochemistry. This is a diagnostic stain for the estrogen receptor. And the estrogen receptor is a molecule that lives in the nucleus of cancer cells, or doesn't. And its presence in the nucleus, where the DNA lies, where the genome is central to its function. So on the left here is an ER-positive tumor, and it's literally just this brown staining showing up in the nuclei of 1% or more of cells. In this case, most of the tumor cells have the estrogen receptor with that brown stain. This tumor on the right is ER-negative. There's a few brown spots here, but these are just normal breast structures, not actually cancer. So because this tumor is ER-positive, this patient is eligible to be treated with anti-estrogens that block the function of the estrogen receptor, whereas the ER-negative tumor, the patient is not eligible for those therapies. So what is the estrogen receptor? It does a lot of things, but the thing we think about most is what's called as a transcription factor. When it's activated by a hormone like estrogen, it moves to the DNA, it binds specific sites in the genome, and it turns genes on or off. And ultimately, this contributes to tumor growth, to therapy response, to invasion, metastasis, et cetera, et cetera. Because tumors that are so-called estrogen receptor positive are so addicted to this signaling mechanism, this activation of estrogen receptor, we can target it with different anti-estrogens. And all of these drugs, tamoxifen, aromatase inhibitors, fulvestrant, and now elacestrant, all of these, and by different mechanisms, target the activity of the estrogen receptor to turn it off, which in most cases is sufficient to stop tumor growth, but obviously that's not the whole story. Mechanistically, though, this is obviously way more complicated. And what I'll talk about today is how estrogen receptor doesn't really function on its own. It always works in complex with a much bigger group of molecules that are ultimately responsible for turning those genes on and off. And my lab has been interested for quite some time now is understanding whether or not the estrogen receptor has different partner molecules in lobular breast cancer relative to other breast cancers, and whether that ultimately impacts estrogen receptor function, therapy response, et cetera. So to do that, a few years back, we got some seed money to do a big experiment to profile the estrogen receptor interactome. Really, we just wanted to know every protein that was physically working with estrogen receptor in these lobular breast cancer cells. So we were able to do this experiment where you use an antibody, which is just a protein that will recognize another specific protein, an antibody to the estrogen receptor, which kind of gently pulls estrogen receptor out of your sample along with everybody that's working with everything that is physically associated with it. You take it out, extract it, and then you stick it into the fancy mass spectrometer box. And this is the magic box you hear about in all of your favorite crime shows that identify every molecule ever. They're good, they're not that good, but we're able to, with mass spectrometry, to identify the proteins and other molecules that come down with the estrogen receptor as part of its partner network, its complex. We did that in a whole bunch of different lobular models and identified over 100 potential ILC-specific estrogen receptor partners, or proteins that were only seem to be working with the estrogen receptor in ILC, but not IDC. A whole bunch of work then by Dr. Sotnick, and a former graduate student in my lab, and others, were able to identify and profile whether a lot of these were functional, what they might be doing. But one particular molecule has become our favorite, and it's this other protein called MDC1. And we've since through a lot more work that I'm not showing you, have developed this idea that MDC1 is really important for estrogen receptor function in ILC cells. What we think about is that in cells that where MDC1, in non-ILC, where MDC1 isn't really working with ER, there'll be a lot of genes that estrogen receptor turns on and off. But since ER is not working with MDC1, there are a whole lot of other genes that other cells don't access, don't use. In ILC, though, we think that MDC1 is allowing ER and lobular cells to access essentially a new suite of genes. It's accessing a new program and turning on the lobular specific functions of the estrogen receptor. And we think that MDC1, as a result, is essentially facilitating the ILC specific genomic functions of estrogen receptor, which importantly, we think include anti-estrogen resistance. So because there's this novel partnership between ER and MDC1, it potentially propagates therapy resistance, but ultimately is controlling what estrogen is doing in these cells. For a lot of reasons, we don't think that MDC1 on its own can actually do that. So we've been trying to understand how MDC1 actually mechanistically facilitates ER control of these unique target genes in ILC and how it's doing this job in ILC. But I keep talking about MDC1, but we do know what it has a normal function. So MDC1 is actually called mediator of DNA damage checkpoint 1. And its main function, its day job, is in DNA repair. And what it normally does is that when a cell detects a break in its chromosomes, which are really bad for cells, they want to fix that, MDC1 immediately moves to that site and acts as a platform or a scaffold for a huge chunk of DNA repair machinery to come in and fix that break. And this is kind of different than what we're seeing in lobular cells. So is it still active? Is it doing its day job? So to ask that, we kind of took a similar approach to what I just showed you for estrogen receptor, and we instead developed an approach to pull down MDC1 and all of its partners. And the idea was that we'd see whether it was working with estrogen receptor and friends, or if it was working with DNA repair machinery. This figure looks incredibly simplistic, but I don't want to undersell how much work Dr. Sotnick had to put into this to make this technique work. Nobody had actually technically been able to pull down MDC1 like he did in this experiment. But then we did that in a whole bunch of models, IDC versus ILC, and sent that back to the mass spectrometer to see what we saw. And excitingly, we saw that in ILC cells, MDC1 was working with estrogen receptor and a whole suite of epigenomic partners, which are the proteins that ultimately are responsible for turning genes on and off. They're kind of the genomic muscle that execute what ER and MDC1 want to do. And that was really exciting because this class of proteins wasn't necessarily what you would expect to see based on what's known about MDC1. We're really excited because among all these proteins we found, several of them are called so-called epigenomic complexes, or groups of proteins that are already known to be partners and to work together. So the fact that we see a whole complex or a group that comes down together implies that that whole group is working together with ER and MDC1, which gives us a lot of confidence about exploring the function of those different complexes. We're excited in part because we think this is an opportunity to target ILC-specific biology, because this partnership of ER, MDC1, and these epigenomic complexes is only working together in ILC. So if we can target it, we can target lobular-specific biology. Importantly, drugs targeting epigenomic factors are already FDA approved. Entinostat is one that's approved in breast cancer. We don't think this drug is going to do what we want, but as proof of concept that this class of drugs is acceptable and is something you can effectively treat patients with in a fairly non-toxic way, it's good proof of concept for us. So we've got our list of favorite targets, and Joe is working very hard to identify which targets we want to chase after and what molecules are our best options to test moving forward. So we think this side of the coin and what MDC1 is doing is a great way to develop new strategies to treat lobular cancer. On the flip side, though, in IDC cells, we see less of this epigenomic machinery, and we see far more of that expected day job, that DNA repair machinery. We're seeing those proteins that MDC1 normally brings to sites of DNA damage to fix things. And while that's really cool, one of the first things we ask is, why aren't we seeing those in ILC cells? Is MDC1 bad at multitasking? Because it has this new job in ILC, which I'll be the first to admit we don't know why it has this new job yet. But does that compromise its old job? And more importantly, can we exploit this to treat ILC? So as I mentioned, MDC1, its primary job is upon a cell recognizing that its chromosome is broken, that DNA is broken, it recognizes it, moves to that site, and acts as a platform or a scaffold to bring in more machinery. And you can literally see that happening in cells. So this is an ductal cancer model where Joe treated cells with a DNA damaging agent, a chemotherapy that induces damage. And all of these little puncta, these little bright foci, these colored dots, represent MDC1 bringing in all that machinery physically to that break site. So all of those little dots represent a break in DNA that the cell is trying to fix via MDC1. So you can see that machinery coalescing in a ductal cancer cell. Lobular cancer cells, we think, are pretty bad at this for some reason that we're still trying to understand. Rather than seeing all of those little points, you just see a red haze. And that red haze is another one of these repair factors that is not quite making it to a site of damage. It's just kind of loose in the cell active, but it's not, the machinery is not getting where it needs to be. And because of this, we think MDC1 might actually be bad at its day job. It's not doing what it's supposed to do to initiate the DNA damage response, maybe because it's busy working with the estrogen receptor, and we're trying to tease that out. Now, what I'm showing you here and all this other work has been in cell lines, in isolated cultures in the lab. So do we think this is actually happening in tumors? And the hope is yes. So the biggest effort to profile lobular cancers to date was via the Cancer Genome Atlas, which was a big National Institutes of Health consortia to profile all of the cancers. In their study of lobular cancer, they identified 3 subtypes, one of which was called proliferative, which is the green line. And patients with proliferative ILC have the worst overall survival and disease-free survival. So these tumors appear to be the most aggressive, and it's about 30% of them. There wasn't a whole lot in the study that defined this subset. The word proliferative was kind of an accident, but one thing that did define them was an elevated DNA repair signature, or activity of a bunch of proteins in one of their assays for these DNA repair factors was elevated. We think these two things are tied together. This inability to efficiently turn on DNA repair is related to what we think is kind of the cells stuck in an on state for trying to fix DNA. And that's what we think the signature represents. So if we use that signature, this elevated DNA repair signature, which I'm calling DDR high here, a high DDR signature predicts worse overall survival in ILC, but is meaningless in IDC in the same data set. So something about this activation of DNA repair is identifying aggressive ILC, and we think it is related to what we're seeing in these cell line models in the lab. Importantly, there are therapies available to undermine dysfunctional or non-working DNA repair. Some of them are FDA approved, like PARP inhibitors. Those are already approved in breast, in ovarian, and other cancers. And there are a whole bunch of trials of other strategies to do the same thing. And obviously, our group is really excited in exploring these therapies as another option for lobular cancer to target both what MDC1 is doing and now to target what MDC1 is not doing in these lobular cancer cells. So really the biggest thing my lab focuses on across the board is that even though we think about ILC as a 95% of these tumors are ER positive, ER is far more than just a positive mark in IHC. And we're trying to understand what that is. We think that the ER partnership with MDC1 in these ILC cells mediates the ILC specific activity of the estrogen receptor, including anti-estrogen resistance. We see in ILC cells that MDC1 partners with estrogen receptor and other gene regulation epigenomic proteins, but instead it partners with its normal DNA repair machinery in IDC. We're excited to try to understand how targeting MDC1 epigenomic partners is one way to exploit ILC biology and develop new ILC targeted therapeutics. And on the flip side, we want to target what MDC1 is bad at doing, what it's forgotten to do, to take advantage of that issue in DNA repair and again exploit ILC-specific cell biology. So that's what I have for you today. We're going to have time for Q&A specifically, but I want to hand off to Dr. Music to talk more about her work in tumorigenesis.
Right. Okay, so I'm Maggie Music, postdoc in Matt's lab, and I'm really excited to get to be here today to talk to all of you and hear what's important to you in ILC research. So today I'm going to share a project that I'm working on in studying early ILC tumorigenesis. So ILC tumorigenesis, first of all, is the initiation or the beginning of an ILC tumor, which starts in normal breast cells. And as you can see here, we have ducts. We have ducts, normal mammary ducts here, right? But as normal breast cells acquire benign alterations and become more proliferative, these cells become ALH, or atypical lobular hyperplasia, or lobular carcinoma in situ. And here we can see there are still ducts, but these ducts are just jam-packed with cells. However, once these cells break through the duct, this is when the cells become invasive, and this is known as invasive lobular carcinoma. So there's no signs of ducts anymore, just the lobular cells are everywhere in the mammary tissue. So although we know that about tumorigenesis, we don't know what is actually happening molecularly. Even still, ILC is treated like any other ER-positive breast cancer with an adjuvant endocrine therapy, mastectomy, or breast conserving therapy or radiation. But their precursors, ALH and LCIS, these are, well, LCIS specifically, although it is biologically a precursor to ILC, clinically it is not considered a precursor lesion, just considered a risk lesion. So this is not considered cancer. However, and the treatment for LCIS is currently maybe surveillance. It's not very clear. However, patients diagnosed with ALH or LCIS have a 4 to 11 times greater chance of developing invasive cancer. So I don't think surveillance is enough. I think that this creates the need to understand molecularly what's happening between normal and LCIS, and find a better way to predict which LCIS will progress to ILC. And one of the hallmarks or defining characteristics of ILC is E-cadherin loss. So E-cadherin in IDC is very prevalent. That's the brown stain, and you see brown everywhere in IDC. However, in lobular, you can see where this red arrow, this stain here is just a normal duct, but the lobular cells are over here, and there's no brown stain at all. So this is E-cadherin negative. So we know that ILC is E-cadherin negative, but actually 70% of LCIS are also E-cadherin negative, suggesting that E-cadherin loss happens sometime in early ILC tumorigenesis. So why do we even care about E-cadherin, right? It's a marker. It tells us that it's ILC. E-cadherin is a protein that spans both inside and outside of breast cells and allows breast cells to communicate with one another. And when this E-cadherin interaction happens, the cell is able to regulate movement, growth, and survival. So there's no overcrowding, so the cells can kind of live harmoniously. However, ILC cells that lack E-cadherin, they're no longer able to regulate movement, growth, and survival in that same manner. So these, the signaling is it's going to change, which all three of these things, the cell's ability to move or to migrate and grow and survive, these are all factors that are critical in the formation of tumors. So how are we studying E-cadherin loss? A lot of research has been done with E-cadherin loss in ILC or ILC cell lines. So these are fully invasive ILC cell lines. However, as I just mentioned, E-cadherin loss happens early in tumorigenesis. So we are using human mammary epithelial cells, which are primary cells, which means they're directly derived from human tissue, typically from reduction mammoplasties. So these are normal, healthy cells. And we are going to knock out E-cad genetically to mimic what's happening in ILC. So we are doing that in these primary cells, HMECs for short. And we specifically have 4 different models of these HMECs. We have 2 derived from premenopausal women and 2 derived from postmenopausal. And so this will allow us to determine the effects of menopause as well, because typically lobular carcinoma is considered to be diagnosed in older women. However, we believe that there's some diagnostic reasons that it's not picked up in premenopausal. So this will allow us to explore that aspect. So we'll take these HMECs and look at how if they are tumorigenic in vivo. And most studies up to today with it's been difficult to study ER positive breast cancer in mice because mice are not ER positive or ER functional. So we are using a unique approach, which is a mouse mammary introductal xenograft model or mind model. And this allows us to inject the HMECs directly into the mammary gland, and this will most closely mimic what's happening in humans. So we can track the transition from something that's pre-tumor to invasive. And we are doing this in collaboration with the Austrian lab at the University of Minnesota. So we'll be going out there to get trained in how to do this mind model, and exchange will train them to do some of the things we do in our lab. So as a recap, E-cadherin loss is critical for ILC. And as Matt had discussed, ER functions differently in ILC. And I don't think those two things are mutually exclusive. So we are, in addition to affecting movement and growth and survival, E-cadherin loss can also potentially indirectly affect ER function. And in some preliminary research that we've done inhibiting that crosstalk with E-cadherin, we have seen changes in ER activity. So we're eager to continue that research from a whole bunch of different angles to see how exactly E-cadherin loss is affecting ER function. So in summary, current ILC treatments have room for improvement. And in order to improve them, we need to have a better understanding about how the normal breast cells are turning into the ILC cells. And specifically, we're asking the question, does E-cadherin play a role in pushing these cells towards ILC cells? And also, what's behind the ER dysfunction or the change in ER function? And what role specifically does E-cadherin loss play in that change? And with that, okay. Any questions about either from what Matt has shared or what I've shared about E-cadherin? Yes.
So that was awesome. Thank you so much, Maggie. I am curious in the age ranges. You have very premenopausal women,
Yes.
Probably nulliparous women, and postmenopausal women. Any thought about adding in a middle age, maybe premenopausal, but multiparous population? Because I know obviously pregnancy has changes on iron tissue. So just want to hear your thoughts about, about that.
We definitely want to do that. And I think once we get these 4 cell lines, when we've got the kinks, I could definitely see that expanding for sure, because that is a great point.
Yeah, I mean, right now we're operating at the extremes for the realities of how much funding we have to do the project. But the next steps are not only using other variable ages, but also ethnic populations. We can get HMECs from other, these are all from Caucasian women. We can get from Black women, from others from the same bank. And then our collaborators at Pitt are developing organoid models, which are more physiologically relevant context to grow these mammary cells, and we can do similar experiments on those organoids once we work up the methodology in these. So the long-term goal is to have a diverse range of models that we do this genetic testing in to see if, is it always the same thing that happens? Is there a specific context that E-cadherin loss does different things, etc. Oh, thank you. The question in the chat is about the connection between CDH1 and E-cadherin loss. Is that regarding, so CDH1 and E-cadherin are the same thing. CDH1 is the gene that encodes E-cadherin. So a CDH1 mutation would cause an E-cadherin protein loss. Did I understand the question right? Can you either unmute or put it in chat? Yes.
It doesn't go the opposite way, right? E-cadherin loss does not necessarily mean that you have a mutation in a CDH1 gene.
Great question. Yeah, that's a great question. So when we say E-cadherin loss, it's meaning that we cannot detect it at the protein level. There are various ways that happens, one of which is E-cadherin mutation. So there are cases in which you will have wild type or normal E-cadherin, but the protein will be suppressed and we don't actually know why entirely. But yeah, so there are different ways that happens. Why do some have CDH1 gene but have ILC? This E-cadherin gene is always intact. That's a totally normal, critical gene for normal cell-to-cell contact. So it's expressed in epithelial cells, which is everything from your skin to the lining of every organ. So you have to have the CDH1 gene to survive. Its loss is actually lethal in mice. But losing it, the cancer cells can tolerate loss for reasons that we don't entirely know yet. That's part of what the point of the study is. What does a cell have to do to accept that it's lost CDH1, and how does that change the cell's biology? Hopefully I'm answering that one correctly.
When you talk about the 3 subsets of lobular, I know when we look at subsets of ductal, luminal A, luminal B, you can actually look at a pathology report and sort of put yourself in that category. Is there anything in a pathology report that tells you what subset of lobular you're in?
No, so unfortunately, the subsets of lobular that I referred to, the existence of 3, there's only 2 studies that have tried to subdivide lobular in the same way with a bigger data set, which is TCGA and a group called RADR. And TCGA found 3 groups and the other group found 2 groups. There's one that seems to overlap between the 2, and it's not clear if it's like a number of does TCGA have more power so they can see more because they have more tumors? Do they see more groups? And how stable are those? One thing that we're really interested in doing is whether we can put together enough gene expression data from collaborators, from public data, to see how robust those divisions are. Like, are there actually 3? Are there actually 2? And then in bigger groups, how do they redefine subtyping? Like a silly technical issue that, there's TCGA has data from a type of expression analysis called RNA-seq, which is a sequencing-based platform, whereas the other enormous data set that we would hope to use a different technology that's called microarray, which is an older technology. And the 2 don't cross over necessarily all that well in this type of analysis. So we're kind of waiting to see how much data we can compile. We, meaning like the whole field, is trying to see how stable those will be long-term. But ideally, we move toward that. Because, we've talked about this a little bit in that PAM50 subtyping that everyone will see, whether it's luminal A, luminal B, that was derived on ductal biology almost entirely, and it superimposes a bit in ways that make sense to lobular, but we really don't have a great handle on how relevant it is to lobular biology. Like, you'll see whether you're luminal A, luminal B, whether that's gene expression or protein staining, but it's not entirely clear how relevant it is for lobular. It is, but to the extent to which is kind of a shrug.
So I have a question going back to the E-cadherin protein, and maybe it's a little more general. So this protein isn't specific to mammary cells, is it, right? And so does it show up in other cancers? Like, are other cancer types studying this? Like, what do we know about it generally?
Yes, I'm not all too familiar with what it is, but I know it is in all.
Yeah, so there is actually a syndrome that's a hereditary syndrome of genetic E-cadherin mutation that is inherited just like a BRCA mutation, and the patients are usually diagnosed with gastric cancer first, and then lobular cancer later. There's actually a lot of misdiagnosis of metastatic lobular cancer to the GI that looks like gastric cancer, in part because of that. So that's the other big field that E-cadherin loss shows up in. I think there's a few other tumor types, but they're quite rare. Whether gastric rolls over to ILC, we don't really have a lot of data on, in part because the hormonal situation is so different. And I could be wrong, but I think the subtype of gastric cancer is not like standard colorectal cancer, so there's not a ton of data on it. But it's something we need to think about, in part because lobular can metastasize happily to the GI. So it's something we need to think more about.
Thanks.
I mean, so one kind of corollary to that is one of the reasons we think that ovarian cancer has some ties into lobular cancer biology is because the way that ovarian cancer spreads in the GI and in the abdomen, lobular can do the same thing. So we're not entirely sure if it's E-cadherin loss that drives it, because ovarian doesn't necessarily lose that the same way, but there's clearly some signaling tie-in that we're trying to figure out what that is. Any other questions online?
One more.
Oh.
When looking at genomic data on CBio, 70% identify with CDH1 and ILC. Are there other 30% a different category?
The 70% with E-cadherin mutation. So most ILC like that one they'll lose E-cadherin by having mutation. So we have 2 copies of all of our genes, remember? You'll have a mutation in one, and the other one, the tumor deletes the other copy of the gene. So you have no functional copies left. That's usually what happens. In the cases that don't have an E-cadherin mutation, they can both get deleted. That's less common. Or there are ways that the gene itself gets shut off, and we don't understand what that is yet. The obvious ways that a gene gets shut off aren't occurring in ILC for CDH1. That was something that TCGA found. Often what happens is the DNA gets a type of mark called methylation that causes the DNA to close up, and then nothing can access it anymore. That's usually how these genes get shut off. That's not happening in ILC. And we don't know what is driving that. It's kind of a shrug at this point.
Can you explain a little bit more about what epigenomic partners are and how they are working in drugs like enostat?
Yeah, the entinostat. So epigenomic partners, what these genes, so when we think about estrogen receptor, what it's going to do, it's going to bind to DNA, and ultimately what it needs to do is physically relax or close the local DNA to allow that gene to be read or not read. The proteins that are the muscle that actually cause the local structure to open or close are these epigenomic partners. What they do is put special marks on the DNA structure that either facilitate its opening or force it to close. Drugs like entinostat is an HDAC inhibitor, and those are histone deacetylases. Histone deacetylases normally work to close up DNA. The thought is that if you inhibit those, you don't allow the cancer cell to turn off things that it wants to turn off. How that actually kills the cell is kind of hand-wavy, but really you're not letting the cancer genome do what it wants to do in that setting, so it can't shut off what it wants to shut off. So we're hoping to take similar approaches. We don't see HDACs, that class of proteins that entinostat targets, in our partners. We actually see more of the opposite. We see, so those HDACs are the deacetylases. We see the acetylases more with HDAC. So there are drugs that do something similar, and in that case, we're hoping that we can stop ER and MDC1 from opening the genes that it wants to open. Hand wavy. There are a lot of things on our list of things to study. Those are our favorite right now. Good questions. Oh, is MDC1 an epigenomic partner? That's a really good question. Our hypothesis at the moment is that MDC1 is reprising its scaffold or platform role that it does in DNA repair, and then it's doing something similar with ER, but it's just scaffolding different things. Hi, Julia. We don't think it itself can do that epigenomic stuff. MDC1 does not have any reported activity as an enzyme that can do that stuff. That said, there's a big chunk of MDC1 in the middle that nobody really knows what it does. So ask Joe next year. Great questions, thank you. All right, well, if anybody has follow-up, I'm always happy to chat. Maggie's happy to chat. So please bug us. But in the interest of time, I think we'll turn it over to Tracy and Donna to kind of kick off the next set of conversations.
Well, hi, everybody. Thanks for coming, and nice to meet some new folks and see some familiar faces. And I'm Tracy Cushing. I'm an ILC patient here in Colorado, and Donna and I are apparently the drivers of the new Front Range ILC patient network.
You voluntold yourself.
Right, yeah, exactly. So I'm gonna have Donna go ahead and introduce herself.
Hi, Sue and Julia. Can you hear okay? I'm speaking from here. Great, okay. So hi, everybody. I'm a patient advocate. I live near Boulder. And I was, maybe I'll just drop down on the slide here. I was first diagnosed with, I'll use air quotes, breast cancer in 2009 when I was 39. And I was told that I had stage 0 DCIS and LCIS. I insisted that they do something with it. And so they removed the breast. And then they said, you're good, have a great life. And so I did, and I have a great life. However, 10 years after that, I had a recurrence. And that recurrence is invasive lobular, but it's also metastasized to my bones. And so, so I'm jumping back into the cancer world. Between that time, one of the things that many of you know probably is that lobular is often difficult to detect. And so even though I had regular checkups, had my mammogram every year, and they would say, okay, you look good. About every 2 to 3 years, they would say, looks a little suspicious, let's do an ultrasound, let's check it out. And then after about 2 weeks of high stress and anxiety, they would say, you're fine, just go back on and do what you do. Except for in 2019, when they said, actually, you need to follow up. So at any rate, since July of 2019 and my diagnosis, I've been on treatment. So once, just in case anybody isn't aware, once you are diagnosed as metastatic, that basically means that the breast cancer has left the barn. The horse is out of the barn, as my surgeon said. And basically at that point, all you can do is treat it and try to stabilize it and stop it from progressing. There's particular lines of treatment that you can follow. None of them are specific for lobular. They treat it exactly like ductal. But one of the challenges is that the lobular doesn't show up very well. So I have scans every 3 months, and they all look great, but I've had progression, even though my scans all look great. So it's a challenge, and so I really appreciate the work that the lab is doing just to advance the science here. So I'm also trained as a physical scientist, and I also am trained in the world of education. So professionally, I work at this interface of science and education. So I'm once an educator, always an educator. So I'm constantly trying to help people understand and raise awareness. So I discovered that being a patient advocate was a great way to do that. And so after the horror of a metastatic diagnosis settles down and you realize, what do I want to do now? I started getting involved in training for patient advocacy. So I actually last year went through like 25 hours of training with the Living Beyond Breast Cancer organization. They have a program specifically for metastatic, which is called Hear My Voice. And they train you in scientific advocacy and policy and self-advocacy and things of that nature. And I've also realized that it's very difficult. Part of the reason that I'm really excited to work with Tracy and all of you to get this Front Range ILC going is that it's very isolating to have a diagnosis because of HIPAA and many other things, they can't connect patients, right? So one of the things that I did is I did a peer-to-peer support program training through MetaViber, which is a nonprofit that focuses more on metastatic breast cancer, to try and provide resources for that metastatic community in the Front Range area. And so we've started that, or I've started that. And so we have this stage 4 MBC peer group on Facebook. And now we're also working to kind of, that's not focused on ILC, that's just metastatic writ large, but about 10% of us, 15% of us that are on there all have lobular. And so anyway, that's kind of my journey, and what I'm hoping to do is just connect people and share information. So much information gets passed between patients that clinicians just don't have any idea about. And so I think the more that we can share and the more that we can connect with researchers and clinicians, we can all be better off. So that's my spiel. I should say too, let me just say, I do also volunteer for LBCA, the Lobular Breast Cancer Alliance, and I forgot to put that on there yesterday because I was putting the slide together really quickly, but I do a lot of just kind of background stuff without not being really super prominent.
Everything you do for LBCA is appreciated, and thanks, Donna. I'm Tracy. I was diagnosed 2 years ago, so I'm still kind of in the thick of the terror, but starting to move past that a little bit. I am a physician. I'm an ER doc. I haven't studied oncology since 20 years ago in med school. But once a physician, always a physician. So as soon as I was diagnosed with cancer, I thought I should probably read up on this and figure some stuff out. I was pretty disheartened to see that research had progressed basically nowhere in the 20 years since I had graduated. I remembered there was this like stepchild, second kind of breast cancer, old ladies, came back in the other breast, something of that nature. That was about it. And then I popped open my pathology book and there it was. And I saw that really not a whole lot had changed. And so I thought, well, perhaps I should get involved. So previous to cancer, I started a podcast for physicians on evidence-based nutrition education because I'm plant-based and I love things about diet and cancer, as well as sort of all the other aspects of cancer that people don't have time to talk about, such as sleep, meditation, mindfulness, community support, food, and stress. And so I'm hoping to move a little bit away from emergency medicine into an integrative oncology practice here in the next couple of years. In my free time, I dabble in triathlons because that's what everybody does in Boulder. So this is a brand new group. It officially exists on Facebook, so now you can join it. And we hope that you will. And we have a lot of ideas and we welcome more input. And this is just almost kind of a spitball list of things that Donna and I came up with in 10 minutes of conversation, and there was a lot there. So in addition to being local advocates for each other and local supports and sharing that, advocacy in the research community, which is one reason why we're here today. Education, I find still there are many women with lobular that really don't know much about it. I love sending people to LBCA's website and I help write their frequently asked questions document and all that stuff because it's the first source of information for so many lobular patients and it's really important. We're hoping maybe we'll have some actual in-person meetings and get-togethers. We talked about also spreading the word on conferences, so the ILC dedicated conference that's coming up in September, but also just sharing about what came out at SABCS or ASCO, or just keeping the lobular community up to date on the latest research from the latest conferences in case you don't happen to be there. And ultimately, we would love to also offer local support to caregivers, maybe children. I also am on the National Parent Council for a group called KESM, which is a summer camp for kids whose parents have had cancer, and it's absolutely amazing. So we're just hoping to kind of share all the stuff that's out there specifically with lobular patients, just so that they can pick and choose how they need support or would like to get involved. Did I leave anything out?
No, I think you covered it. The world is our oyster at this point.
Exactly. And we need help and we need all of you to join. So look out for us on Facebook and please join us. Now I'm going to hand it over to Lori.
All right, thank you. It's really nice to be in Denver. It's a little cold, but beautiful snow in the mountains, so it's nice to see that. First, I want to say a little bit about me, because one of the great things about advocacy is we are all so unique. There's not like a job description and you fill it out and we all have the same background and characteristics. We all have very different backgrounds, and that's what makes advocacy, I think, so strong, is the diversity of the group of people who want to be advocates. I was diagnosed in 2010 with stage 2 pleomorphic lobular. When I went to Dr. Google, there was absolutely nothing about lobular, or very little. And there wasn't the social media support groups that we have now. All we had was breastcancer.org. And I did go there, and there were lobular patients there, and it was very interesting. But it took about 5 years before there was a posting on bc.org that said there's going to be the first ever International Lobular Breast Cancer Symposium in Pittsburgh. And it was a patient advocate who posted it, and she said, I want to invite patients. And I went. And I know Julia is on the call, and Julia Levine and I went together from Los Angeles. And that's where, it's the first time I heard a lot of talk about lobular. It was way over my head. So I just kind of wrote notes of buzzwords I wanted to look up later. I came home and I looked at my notes and it said, I lost my E-cadherin. And it was the first time anybody had talked about E-cadherin. So there were a group about maybe 8 or 10 of us from that first symposium of patient advocates who formed the Lobular Breast Cancer Alliance. And we formed it under the leadership of Dr. Steffi Oosterich, who was the chairperson of the symposium, and Lee Pate, who served as the advocate head. And I'm happy to say that the Lobular Breast Cancer Alliance today is still very strong. It's the go-to source for information on lobular, and I'm really proud to have been a founding member. I have trained myself in research advocacy. It's an area of interest of mine. I've been to the MBCC project lead and tried to attend as many scientific conferences as possible. And I'm currently engaged in a few definitely basic translational and clinically focused research aims. So I'm working with Matt on a couple of his projects. They're not all lobular. I am on a translational research grant that is involved with the microbiome in breast cancer. It's really interesting. I'm on a DoD grant for a clinical trial that is cryoneurolysis for mastectomy, post-mastectomy pain instead of opioids. And having had 2 mastectomies 8 years apart, it was very near and dear to my heart to be involved on that. So, you know, advocacy involves a lot of different things. I know you all know that. Peer support, we've talked about that. Donna has some experience there. I do mentor for ABCD and Share Cancer Support. There's research advocacy. I know education and awareness. Tracy talked about how important that is. Public health policy. Right now in Congress is a bill called the Metastatic Breast Cancer Access to Care Act. It's not up for a vote yet, but we are hoping that it will be up for a vote within this current Congress, which will be this year and next year. And when that happens, we all need to write to our congressman and say, please vote for this act. So public policy, it's something as simple as an email or a phone call to your congressman can really help. And fundraising, obviously, to get research, we need fundraising. Yeah. So why should researchers involve patient advocates? Well, as an advocate, we ensure that the focus for the research is on the patient, but it also adds that human face. And I've met a couple of researchers and they'll look at me and they'll say, I've never talked to a patient before. And it really changes their sense of urgency. You know, there's the human face behind what they're doing. It stimulates discussion. They love to ask me questions about my experience, my hopes, the pitfalls I've gone through. And I love to talk to them about their research and why they're there and why they're interested. And, you know, as I mentioned, because advocates are so different, it really provides a diverse perspective. The more advocates they meet, the different their perspective will be. And these are the things that I look for when I'm asked to be on a research grant. And you can never ask an advocate too early to be on a grant or to be involved in some type of research activity. First of all, specific opportunities. Are they looking just to have a letter of support? Are they hopefully looking to have some impact into a lay abstract to write for a grant? Are they interested in disseminating the information to the community once the research is done, whether it's good or bad? I need to see clear expectations. I have gotten those emails where I have a grant due tomorrow, would you write me a letter of support? And it's like, no, I want the opportunity to read the grant and then talk to the researcher, because I'm not a scientist and I rarely understand the grants unless I can talk to them and ask them to put it in lay terms and ask the questions of why it's important. So clear expectations are really important. And then providing the meaningful participation. You know, I don't just want to write a letter of support. and then walk away. I want to get updates, quarterly updates of what's going on. And because I'm not a scientist or a researcher, I like to ask questions, and there are no dumb questions. And I'm sure I've asked all the dumb questions, but I need to. And these are the skills that I've tried to put together in my toolkit to make me a successful research advocate. First, I just want to make things better. I want to address breast cancer. Of course, lobular is near and dear to my heart, but really, I think the entire issue of breast cancer needs to be addressed in order to even address a little subset. Strong communication skills, particularly listening. At these conferences, it's very easy to just kind of become glassy-eyed for me when you hear so much of the science talk, but you find those buzzwords and you kind of click on them and you just ask the questions. It's always okay to say, does this mean that, or can you give me an analogy of something I might be able to understand? I also think it's really important as a patient advocate to represent all patient stories. I mean, my story I think is really important. However, I have a lot of friends and a lot of colleagues with different stories than mine, and it's important to represent the entire group. Willingness to learn, sometimes that can be really difficult, but I love particularly working for the DoD as a consumer reviewer, because I get those grants and I'm like, oh, wow, I got a lot of work to do. But you just continually learn, and I do find it's very interesting. The changes in treatments and protocols. So I was diagnosed in 2010. Things are a lot different now. So when I do peer-to-peer support, I learn a lot from current patients about surgical options, imaging options, treatment options. And I think that's really important in staying abreast. Working collaboratively, obviously nothing happens in a vacuum all by yourself. And then probably most importantly is adaptability. So things can change. During the course of research, the hypothesis might have been found out to be not true. One of my grants, the microbiome in the breast, that in fact happened. And we had a quarterly meeting and the researcher said, it's not true. So we're going to pivot and we're going to take this because now maybe we think it means this. And it was really interesting. So everybody has to be adaptable. I mean, science is not always predictable, as we know. And that's all I have to say. Thank you. Who's next?
There's a Q&A. Jade, you're gonna ask a question?
Yeah, I was interested in becoming a patient advocate. I'm newly diagnosed and I'm somebody who likes to learn a lot about things. I've probably deep dived into this by reading about, 200 or 300 scientific journal articles that are way above my pay grade. I feel like I could get into advocacy. What would you recommend that I start? Where would I start? Something like that.
Great question. First of all, there are great resources out there. The Lobular Breast Cancer Alliance website has a resource toolkit, and there's a list of things because there are some online classes you can take through the Research Advocacy Network that teach basic science of cancer. There's also, different organizations. The National Breast Cancer Coalition does a leadership summit, as well as Project Lead. The Young Survival Coalition has training. Living Beyond Breast Cancer has training. I'm not sure where you are, where you're treated, but there could be local advocacy groups within your organization. But I would really look at the toolkit on the Lobular Breast Cancer Alliance. There's also a comprehensive resource on the Cornell website. It's put together by a male cancer patient advocate. Gosh, I have a link to it. It's probably like 5 pages long. And he includes the LBCA on there. And maybe please sign up for our newsletter. I'll put a link to it in our next newsletter. So
I'm not sure where to sign up for the newsletter.
Okay, on the Sikora Lab.
Yeah, it's sikoralab.com. There's an
Okay.
advocacy tab. There's a link there.
Okay, great.
I'll throw out too that we are kicking around doing like a breast cancer / cancer boot camp this summer ahead of the Pittsburgh symposium, to kind of help with getting basic science up to speed so you can follow some of the science in Pittsburgh. No plans concrete yet, but we're kicking around the idea.
But there, the Research Advocacy Network is online. Their classes are free, and even like for Project LEAD, which the National Breast Cancer Coalition does, before you go to Project LEAD, they want you to go to the Research Advocacy Network and go through their classes. So it's really a great grounding in understanding. And then if you're interested in working with researchers, that happens just by kind of meeting them. If you go to Pittsburgh or local conferences or meetings, or see a research study that you like and you look at the researcher and he happens to be in your town, you call them up. I mean, they're really open.
Twitter, that's how I discovered.
Twitter, that's right.
So there's a huge community on Twitter that's a combination of patient advocates and oncologists and researchers. And so connect there, and I saw, Lobular right down the road from me. So use social media as well, like to find researchers.
So our next speaker is Jen Lowry from the University of Colorado Comprehensive Cancer Center, the Office of Community Outreach and Engagement. She's going to talk to us a bit about what COE is working on to involve disseminating information to the community, and importantly, the Cancer Center's efforts to build a patient advisory council. I think the acronym is CARES. I don't recall what it stands for. But thanks, Jen, for being here. We're excited to hear about what the Cancer Center is doing. I'll hand it off to you.
Yeah, no, thank you. And thank you for whomever would help me advance my slides.
I will, yeah.
I'm Jan Lowry. I'm at the in the Office of Community Outreach and Engagement at the University of Colorado Cancer Center. I'm pretty new to this role in the Office of COE, but COE itself is relatively new, and I will talk a little bit about that. Just by way of background, I'm a cancer epidemiologist, and I myself have done a fair amount of my own research with breast cancer and with colorectal cancer, focused primarily on cancer prevention. So I have a faculty appointment in the School of Public Health as well. So again, thank you so much for the opportunity to be here. If you could just advance my slides. Here's our team, as Matt referred to, the COE team, as we call it. The director or the associate director is Evelyn Orayo. She also has a faculty appointment in the School of Public Health. We have a small team, but we are beginning to grow. We've already added one more member to our team, but this is the group as is today. Next slide, please. So I thought what might be helpful, not knowing what everyone's background knowledge was, and certainly, as I mentioned, the Office of COE is pretty new to me. It's a new role for me. And like I said, it's pretty new on the landscape, right? Just for cancer centers in general. So I thought it might be helpful just to give a backdrop of what COE is and how we came to be. So University of Colorado Cancer Center at Anschutz is one of, it's an NCI designated cancer center. So we receive funding from the NCI. And a few years back, the NCI came out to all the cancer centers and said, and this is in 2012, we now expect that you're going to sort of expand your catchment area. So you can no longer refer to your catchment, if you will, as patients who live in your, the communities that directly surround your institution, nor those that come to your front door. You have to define a catchment area and expand that, right? So for us, our catchment area is the entire state of Colorado. And then really they came and said that you have to that is your service area. Those are the residents that you serve. And then 4 years later, they came out and said, and by the way, you need to have a community and outreach and engagement office. And that comes with a number of requirements as listed below that were just updated in 2021. So essentially what they're saying is that you now have to go into your community and engage them in everything that you're doing, right? So you have to define your catchment area, which is, as I said, the state of Colorado in terms of cancer burden, incidence, mortality, late stage diagnosis, understand the demographics and understand where those disparities might lie within your catchment area. You have to perform research across this cancer spectrum. As Lori was mentioning earlier, this is basic science, clinical research, translational research, and population-based science as well. And importantly, you have to engage the catchment population, right? We can't just be doing this in a silo. We have to be going out into the community, getting input from our community members, bringing that back to researchers at the university so that the research that we are doing is really targeting issues and concerns within our communities. We have to design clinical trials to represent the diversity of the catchment population. That is very much a focus of the Cancer Center right now, to increase community enrollment into clinical trials, translate research into policy recommendations, and then extend research and policy both within the catchment area and beyond. So again, I was at the Cancer Center for many years. I left and I came back, and all of this sort of is pretty new, and we're all still trying to, get our head around it. But I thought it might be helpful to understand why COE is and what all we are charged with doing. Next slide. So our office has 3 aims. One is to monitor and disseminate data on, again, cancer burden, risks, and disparities within our catchment, which again is the entire state of Colorado. Second aim is to facilitate community engaged research across scientific programs and clinical trials. So that's sort of bi-directional, is connecting investigators in the Cancer Center with community, but then also connecting community back to the Cancer Center. And then the third aim is engaging communities in disseminating and implementing cancer prevention, evidence-based cancer prevention initiatives in their own communities. So when I talk about this, I think of our role as sort of going into the community, helping build their own muscle around doing this work in their own communities. Next slide, please. So this is just an example of under aim 1, again, monitoring and disseminating data. We work with PHSR, which is a group within the Cancer Center that helps us with a lot of the data, the catchment and visualization. So on the left, for example, we have highlighted or visualized cancer disparities. So the darker colors, the darker blue and the darker orange, represent rural or frontier counties in Colorado. Blue represents high cancer disparities, so high cancer incidence, high mortality, and high percentage of late stage diagnosis of cancers. The lighter orange and the lighter blue represent the urban counties in Colorado. Again, the blue represents those high cancer disparity areas. So on the left is all types of cancer. On the right is lung cancer specifics, right? So again, the darker green and represents sort of the higher cancer disparities, higher late stage diagnosis, higher mortality, et cetera. And I'm showing this to you because we use this both out in our communities to help those communities understand the cancer burden in their communities. But then we also bring this back to the researchers within our in our Cancer Center to help them really understand what those disparities are and how they might focus their research. Second slide or next slide. So second aim is to facilitate and support the research going on at the University of Colorado. Facilitate is sort of a key word for the Office of COE. We are not necessarily doing the research ourselves, but we are facilitating, again, in this with aim 2, sort of the connecting the community with the researchers. So here's all the different services or activities that we're involved in within COE. We do clinical trial education. So we're just finalizing a video that we're pushing out to the community and hopefully bringing it back into the clinics. So at UC Health to help lay people of all backgrounds sort of understand or demystify what clinical trials are and what they aren't. We provide English and Spanish translation for consent forms or other study materials. The Patient Advisory Council, which I'm going to talk about a little bit more here today, which we're right now calling COE Cares, but it is a work in progress. It's not established, but this is something that we're really moving toward in the next few months, putting together. We do Spanish-speaking patient navigation right now, mostly patients from Denver Health, but we're going to be expanding into other FQHCs around Colorado. Community outreach and engagement to enhance enrollment of community members. Again, sort of that bridge, facilitating community with researchers, making sure that the research they're doing is focused on community needs. And then our team is, we do a lot of this sort of in tandem or in partnership with the Cancer Coalition with the state of Colorado and also many of the statewide task forces. Next slide. Briefly, third aim, I know that's not the focus today, but just to give you a flavor of some of the things we do to engage communities in disseminating prevention and control activities. We're partnering last fall and this spring with 365 Health, which does health fairs across the state of Colorado. Again, the target population here at the fairs are people that may not have a medical home, that are in need of that is their annual way to get their typical screening, including cancer screening, and we're handing out colon cancer fit kits, screening tests at these fairs. We often go to community events, provide general education about cancer prevention and control, and again, try to support communities in their own efforts in doing so. I've mentioned our partnerships with Statewide Cancer Coalition, and the last thing is and I mentioned it before, maintaining and establishing this bidirectional communication. We do have what's called a community advisory council, just to get these confused. And these are community members representing diverse groups across the state. We meet with them quarterly, so it's separate from the patient advisory committee, which I'll be talking about shortly. They come together. We share with them information about the catchment. They help us as a team and a cancer center guide activities on a more broadly across the cancer center. Next slide. So just coming back to this is sort of under our aim 2, this patient advisory council. We have come up with the interim name of COE Cares, Community Advisors for Research Equity in Science. We're calling it a work in progress because we actually would like, once this is established, for the members to name themselves, if you will, right? With what they think is most appropriate. And the purpose of COE Cares for our Patient Advisory Council is to help investigators at the University of Colorado essentially do community engaged research. And they would do this by providing input and feedback on study materials, study design and methods, participant enrollment, recruitment into studies, including clinical trials, and importantly, and I heard this from Lori, how to actually bring your results back to the community and share it back with the community that it might be impactful for. Our vision is that the members will strive to provide input and feedback to investigators to improve community engaged research, which ultimately leads to improvements in patient outcomes. And importantly, again, back to this catchment idea, to ensure that all persons in Colorado are represented in research. Much of the work we do, we align with the Colorado State Cancer Plan, and just as an FYI, establishing this Patient Advisory Council does clearly fall under one of the strategic objectives, if you will, of the Colorado Cancer Plan. Next slide, please. So in terms of scope of work, and again, this is a work in progress. We have a draft charter that we put together. We envision that our community advisors will participate in meetings and with individual grant projects with investigators, with other members of their research team, other cancer community advisors, and cancer center staff to discuss catchment-related research projects, to review cancer-related protocols, grant opportunities. I know it was mentioned on the previous presentation, some of the grants now, DoD for sure, require that investigators have these patient advisory committees. I think that's where NCI is going, and I think it's going to become more common, but that's a requirement. And that our advisors would help sort of provide feedback on other study-related documents. Right now, we're thinking the meeting format would be monthly or quarterly, and honestly, that really depends on demand. I think a lot of work initially is going to be advertising, if you will, about the work and the service that this community advisor group can provide to investigators. We're envisioning a hybrid format because we would like our members to be representative of our statewide population. So we understand that some folks will not be living in the Denver metro area. And we're envisioning, again, maybe 1 to 2 hours per meeting. We plan to compensate our members either per meeting or hourly, working through that right now, and then to reimburse for any travel expenses as well. Next slide. So in terms of membership, I think we're, again, trying to be really deliberate and thoughtful about members, not to be exclusive, but understanding that we probably would maybe max at about 10 to 15 members. Again, I would love feedback from this group on that. We want our members to reflect the diversity in culture, race, ethnicity, gender, age, and healthcare experiences. So this would be uninsured, insured, et cetera, of all Colorado residents, right? Again, we have to really be representative of our catchment area. We would require that members complete online training for human subjects research, and then we would encourage them to do other training as desired. There's a lot of programs out there that offer all different types of training for individuals, and we thought we would offer that as a benefit. We're thinking it might be a benefit for some. And then in terms of experience with cancer, I think that's something that we would like in terms, but folks can be either cancer survivors themselves, family members of survivors, caregivers, healthcare providers, or health educators. In terms of the membership at the Cancer Center, of course, COE, our office, we're leading this effort. We've just hired a new coordinator that's going to take this on, at least part-time, help with this up and going. We will also facilitate meetings, distribute materials to the members, et cetera. And then, of course, Cancer Center members, we would rely on them. I mean, essentially to show up, right? To submit proposals and say, hey, we have this grant, or hey, I have this need, or we're, whatever the case may be. But again, I think a lot of work is going to be needed ongoing to remind investigators of this wonderful resource and absolutely the true benefit of utilizing this group. So I think that's all I have, Matt. I think that the last slide is just really a slide to give you Evelyn and my information. We are, again, trying to finalize. We're at the stage where we're finalizing the charter. I think our next step is then to really put a plan together of how to, quote, recruit patient advisors that would like to be part of this, this program. So I think I'll stop there and maybe answer any questions.
Thanks, Jen. I think that was really great. I'm excited for what the Cancer Center is trying to do.
Do you imagine that the Front Range advocacy group that you're putting together falls within their organization, or is going to be separate from them?
Separate, but I think it'd be great if we can have at least one member of the group that participates. I think it'd be great to have an interface with the Cancer Center with CARES. That was part of why we invited Jan to talk to us today.
Yeah, and something that's come up in our discussions, Jen Richer, who's a researcher, I think she has a DoD grant. She has so there's almost like two different levels. There's this monthly or quarterly meeting that everyone participates in, but we expect that some investigators are going to want to pull away or ask for help from specific individuals to do more of the granular work, right? And maybe work more closely. And as Lori said, like way upstream, like even in conceptualizing the grant and who's the target population and all of that, it might be that there would be some individuals that would maybe want to work more closely with investigators. So for us we're thinking through, maybe we have a set of 10 to 15 that are all the time. And then we have a larger roster of folks that we could tap as they align their interests with certain investigators. Does that make sense?
Yeah, and if I can jump in, I think that this is a potential role for the Front Range ILC group, right? Is that we could be a resource for that. Like if you're looking for people, particular demographic or experience or whatever, then we can kind of help identify those people and point them in that direction.
I think that's great. I mean, I'm not going to pretend that my research is going to cover all of your collective interests. So I think it's fabulous to plug in as a resource in that context. So Jan, at what point are you in actually recruiting the main board versus the broader, research advocate roles? Do you want to hear from folks today that want to get involved? Where are you at?
Yeah, I mean, good question. I think if anyone, well, first of all, I mean, just watching the earlier presentation, there's a lot we can learn from you all, just in how to set this up and even with our charter and just making sure it's successful in advertising it and demonstrating the value. We could learn a lot from you all. So it would be really nice if anyone's wanting to join us and just help us. So that's a totally different level, but just kind of join us and help us set this thing up and kind of walk us through the finish line. And then maybe, I mean, we would appreciate any ideas folks have about how do we recruit folks for this? Like, how do we do that? What's the best way to do it? And then anyone interested in maybe just joining the CARES group, that monthly or quarterly group, and then folks that just want to be maybe on I call it a roster, it sounds horrible, but just like a list of folks that really want to do this, but maybe they have very specific interests. Like I want to work on projects where it involves, as you're mentioning today, just ILC or breast cancer. I think that would be helpful. And I can follow up this call, Matt, with maybe a survey you can send out to the group, and then they can just respond and say, this is my level of interest or not.
I think that sounds good. I see nodding in the room. So yeah, that sounds great, Jim.
Okay.
Can I add one other thing that I was actually really excited to see, Jan, is that for whoever's on this CARES board or committee, that you're offering some compensation and travel costs, because especially, and this is, probably preaching to the choir, but especially if you're trying to really reach diverse groups. One of the things that is kind of known, especially in patient advocacy and a lot of other things, is that the people that have the hardest time being able to participate in this are the ones that, struggle financially or don't have the time or, are working multiple jobs. And so I was really pleased to see that was a component, because it's really important to be able to get that diverse group recruited and involved.
Thank you. Yeah, we want to honor that. I completely get what you're saying.
I've raised that a few times in Canada. From a Canadian perspective, can you hear me?
Yes.
Okay, good. And usually it's met with crickets, like nobody wants to comment on financial compensation for patient advocates. And I did participate in an ASCO lobular breast cancer grant, and it was a lot of work, and everyone else is getting paid through their job, but the patient advocate isn't receiving anything other than the prestige of doing it. But it's I actually had to go pay to print out the grant so I could read it instead of reading it online. So there's those incremental expenses that may occur for patient advocates too, that should be considered in the whole process.
Following up on that and talking about partnerships, I think I saw Sienna online. Oh, great. So Sienna Kostner is the manager of strategic partnerships with ACS North. This group has been a fabulous partner with me in helping support my research. But I'm eager to hear her thoughts on how ACS can help support this group as well.
Hi, everyone. Thank you so much for inviting me today, Dr. Sikora. Since talking, I do have a new role in addition to the Colorado Research Lead. No worries. In addition to being the Colorado Research Lead, I am the Associate Director of Community Partnerships over Colorado and Utah, focusing on FQHCs in Colorado and Utah. So it's been a while since we talked, but no worries. I'm just going to give you guys a little bit of a background. First of all, we are honored to fund Dr. Sikora's research. His research really exemplifies a lot of what we look for at ACS. And he, especially with his dedication to the ILC patient population. So thank you for having me today. To give a little bit of context and background as to what ACS does, we are the largest nonprofit, or pardon me, the largest non-governmental funder of cancer research in the US, with funding over 58, can you hear me? Can you guys hear me?
Yep, all good.
Okay. With over $5 billion in research funding since the 40s, we very often fund the first research of young researchers. And historically, 50% of ACS research has gone to women as well. We have funded 32,000 grants, more than 25,000 investigators, and over 1,100 institutions. With that said, we fund currently 19 researchers in Colorado. So where we focus our research, we focus on 6 research priorities that span across the cancer continuum. 1, focusing on causes and etiology, obesity, healthy eating and active living, screening and diagnosis, treatment, survivorship, and health equity. All research really, all proposed research fits into one of these major priorities. So here we have, here I have some more information right here. Sorry, I lost my page. So here we have some benefits of our ACS applicants and grantees and how we really support them. So our peer review system is really designed to help the applicant improve their science, even if we don't fund it. So they are given a community and they give feedback as to how they can improve their grant. If they were not funded, how they can improve their grant and become better at writing their grant, expressing their grant. And so that's a benefit of the applicants. Fostering a mentoring atmosphere during that review meeting to really just have the researchers at ACS to fall back on to say, hey, let's focus on this. Let's really, really highlight this area. And so after that, the scientific reviewers provide constructive criticism for the applicants so that they can build and become better with really explaining their research to other communities. And then the grant mechanism for young, mid-level, and senior investigators really helps to drive their careers and not only be funded at the lower end, but also have opportunities to be funded through Discovery Boost grants or Mission Boost grants, Clinical Scientist Development grants. So these are postdoctoral fellowship grants. There's a lot of different opportunities for them to be granted throughout the lifespan of their research. For actual grantees, opportunities for further funding for established grantees, as I just mentioned, provide networking and mentoring opportunities for young researchers with distinguished ACS professors, so they can have a mentor with one of our executives to really boost their research. And also, not to mention, the postdoctoral fellows have a generous parental leave policy as well. So we at ACS really like to look at the family and work balance as well. With that said, we are very, very excited to help support Dr. Sikora and his efforts with this ILC community. We are still looking and open to ways that we can partner in the community and provide resources. So we are open to that. We have gone through some reorganization where we can also co-brand. So if you guys have information on how you want to co-brand in the Colorado region, reach out to me so we can talk about those logistics. But really just looking at how we can help support everyone's efforts in the community, whether it's providing resources for you, coming to events, and especially with Dr. Sikora's research, just being available. Do you have any questions?
So I thank you, Sarah. That's very interesting. I had no idea that that ACS did this. The things that you were talking about, is that primarily in support of the researchers and the grants like are on the slide? Or I'm just thinking like, as we're developing this new community in the Front Range, are there opportunities to partner with ACS for resources or pointing people to things, or do you have patient advocate resources or community resources in general that we can tap into?
Yes. Definitely. We are looking at partnering in community with your group. And so I'm just the research lead for Colorado. We have an extramural discovery team for corporate that oversees everything. But my day-to-day job is to work with FQHCs and provide resources for them to help their patients. So we have toolkits, we have different resources such as Road to Recovery if they have transportation needs or lodging needs. We provide grants for patients, looking at how we can best look at the journey of the patient from diagnosis to treatment and survivorship. We provide that information for medical providers to talk that through with their patients as to what to expect. We even go as far to talk about what to expect for survivorship. How much does it cost for survivorship? So we do have a lot of different resources available that we can provide at the patient level as well.
Okay, great, thank you. Looking forward to figuring out how to connect that.
Definitely, thank you.
You're in charge.
All right, well, welcome back. Sorry, we were chit-chatting and coming up with ideas and brainstorming, but that's kind of where we're at right now with the Front Range ILC network. We're very young, very new. We will send you all a Facebook link. We want you all to join, and this is just a few of the things that we came up with that we thought we might help work at. Obviously, we all have a good relationship with the Sikora Lab. Matt has a roster of advocates he can probably reach out to now, but we can certainly share that love with other research groups that might need ILC advocates. We would like to help educate people about other advocacy opportunities. So all the things that Lori mentioned, we'll keep circulating information on training, education, other resources for folks that want more, want to learn more. The ILC symposium in September will be a great opportunity for us to advertise a conference, get people either to attend virtually or in person, and certainly provide as much follow-up as we can on what's presented there, and in a way that's accessible for all folks. We talked about maybe doing some in-person fundraising, some 5Ks, 10Ks, other events. I'm doing triple bypass to raise money for LBCA this year. So anything of that nature that we can combine forces in the Front Range, there's certainly no shortage of things. And eventually, as I mentioned earlier, we'd love to get resources together for families, caregivers, kiddos, and all the other people that are affected by an individual's cancer.